Sorry, what do you need?
I've submitted, well, I've submitted to our committee, and they have
Whatever you're active for. You guys do this.
I can't even get to the
Regularly years from now.
Yes. What?
Do you want to type it? This is your name support for email. That's okay.
Okay, that's right. Yeah, I have a couple letter records.
Thank you very much.
Have the several,
Let's get you checked in if that's okay. This new department of email.
Really? Yeah. Oh my gosh. Yeah, so this is the camera here.
Okay.
You see, it's this is, you can kind of see it here. It's got a picture of the screen, so if you stand right there, this light should turn. It's not yet. Maybe it's live already. Let's check.
Hello. I think I missed you checking in, if that's okay. Please. If it's easier, I can get it to you if you want to do it. That way I don't mess it up. Sorry, it looks like we're working on it live slide. Okay, sure.
We'll start live soon, and then Yuri will introduce. Yeah, it's just that camera with the little red light.
Oh, got it. Fantastic.
We should start, but I gotta find Sabina to start this. Oh shit.
Thank you.
She says it's live. Okay, so I guess it's live. Yeah, that's right. Here you go. Just adjust.
All right, good afternoon, everybody. It's my pleasure to introduce our speaker today, Luke Edelman from CS Genetics, another company that has a pretty cool technology for single-cell RNA-seq and other single-cell approaches. Luke is, his undergrad was in the University of Illinois, followed by a PhD in Cambridge, and now he's CTO and founder of this company. As you all know, single-cell sequencing became pretty routine lately. You can't even call it cutting edge anymore. Everybody doing it left and right, but it's still very challenging in some cases and still very expensive. And so I'm very excited about this technology because it doesn't require any microfluidics, doesn't require any instrument. It is not sequential barcoding. There is no emulsion PCR involved. So you will hear how it works, and I think it has a good potential. We are going to implement it as a service, hopefully, in our core. But the beauty of it is that actually any lab can do it, and this is what I'm hoping for. So Luke, it's all yours.
Thanks, Yuri, and thanks to all of you guys for coming around. I have a few slides to show you guys today about our new platform for single cell that was just launched at the end of last year. So if you're new, Jeff, can you hear me?
Loud, a little loud.
Louder? It's rare that people tell me to speak more loudly. But
If you want to use this microphone.
sure. Is that,
Turn it on.
you guys hear me?
Yep.
Is that better?
A little higher.
All right. Is that good?
Better.
All right, fantastic. Well, thanks a lot, Yuri, and thanks to all of you guys for your time today. We're walking through a new platform for single-cell omics that our company, CS Genetics, just released at the end of last year. As Yuri was saying, it's a pretty different technology platform compared with others in the single-cell space that you may be familiar with. But we think it has a variety of practical and technical advantages that could be attractive for a variety of applications that you may be using right now or that you might be considering going forward. So we have a fair number of slides to get through, including related to the technology itself, some early customer data, some things about our workflow. Please feel free to interrupt me at any point, but we'll have plenty of time for questions at the end. So we'll cover our new platform for very simple, scalable, and flexible single-cell library prep, which we're commercializing as the Simple Cell platform. Now, our company as a whole has a goal of philosophically making single cell more accessible, more democratized, basically like less scary than it generally is. I think on average, when people do single cell today, it's in a very tightly controlled experiment where you're worried about the money, you're worried about the workflow. We'd like to make single cell a much more regular and low stress kind of bioscience experiment than it generally is today. Just to walk you through a little bit of what's backing up our technology and the products that we're going to show you. A lot of it comes down to some pretty significant, what we think is new substantive innovation in single cell. Our tech works in a very different way compared with other technologies in the single cell space. And as a function of that, we have a pretty big patent estate. And for those of you that track the business side of single cell, there's a fair amount of lawsuits and litigation that goes on back and forth, which you ask me in a personal sense is a little bit of a bummer, but as a company, you got to make sure that you're ready for that stuff. And a big chunk of why we're excited to have this platform now on market is that it's a very different platform. We don't see ourselves as being exposed to the patent and litigation threats that others in the industry have been making. Anyways, onto the fun stuff. We have new kits based on our new technology that we call kinetic confinement for simple cell gene expression. So basically a 3' gene expression assay, very similar in terms of the kind of data that you get from other 3' gene expression assays, but with a very different biochemistry underlying it. So before we get into the technology itself and some data, this is basically what the kit looks like. As you already said, it's an instrument-free technology, so it's all delivered just in similar to an NEB or a QIAGEN box, just 2 boxes. There's no special equipment or instrumentation that's required. It's basically our boxes and then standard PCR machines, ethanol, other stuff you almost certainly already have in your lab for other workflows and experiments. It's by some margin, we think, the simplest workflow in all of single cell. It basically feels like a normal bulk library prep assay, but it's a single cell assay. And it has a variety of flexible pause points across the workflow that we'll describe in the coming slides. It's very automatable, and it has very low sample input requirements. So if you have small samples or precious samples where you don't want to waste all of the material, or you're otherwise concerned about not having enough cell input on a per sample basis, it's a pretty good platform for those kinds of applications. And lastly, it's very flexible, very scalable. So as you'll see, we have kits of a couple of different sizes, 8, 16, 96 samples. But something that's unique about our platform is you buy, say, a 16 sample kit, you don't need to do all 16 samples at once. You could do 4 samples or 8 samples at once and keep the rest of them for the next day or the next week or whatever. And that's a flexibility that we think is actually important for how people would want to use single cell, but generally can't with the other platforms. So to get into a little bit of how the tech itself works, So it's entirely instrument free. It's all done in normal PCR tubes or plates, depending upon the scale of your experiment. A couple of other just top level points. It's a single sample in a single tube carried all throughout the workflow. As you already said, there's no combinatorial indexing. There's no steps of pooling and then re-splitting your samples. So if, for example, you have one sample that maybe was dodgy, it doesn't affect the other samples. So there's an independence from sample to sample across the entire experiment that we think is useful as a sort of functional feature of the platform. So going into a little bit of how it works specifically, to give you guys a conceptual context here, any sort of single cell platform, be it ours, be it those that are instrument-based, be it the other instrument-free platforms, have to address the same fundamental technical challenge, which is taking your sample of cells, labeling the nucleic acids of cell 1 with index 1, those of cell 2 with index 2, so on, so that you can pool all of those labeled nucleic acids, put them in a single lane or a single flow cell of a sequencer, and then informatically deconvolve all of the nucleic acids that came from cell 1 because they had index 1 appended, those from cell 2 because they had index 2, etc. The way most other single cell platforms work is by partitioning, meaning the way you achieve that single cell indexing is by taking your sample of cells, subdividing it into a large number of partitions where you have your index sequences, and then inside of those partitions is where you do the single cell indexing reactions. We've, in a conceptual sense, basically turned that approach on its head, and instead of taking the sample of cells and subdividing it into the index sequences, we load the index sequences on a new type of bead and bind those beads with the indices directly to cells just at a PCR tube. And that's what allows us to do what is essentially bulk liquid handling, but give you single cell data at the end. So the key steps of the workflow up front are all labeled here. So there's a new type of cell reagent pairing reaction driven by our new type of indexing reagent bead. And there's a new type of solution phase cell indexing reaction driven by something that we call kinetic confinement. So I'll just walk you through each of these in sequence. The indexing reagent beads that we use are similar conceptually to like the gel beads that you may be familiar with from 10X. Each bead has millions of identical copies of a given index oligo, but the different beads have different index oligos. So like the red bead will have millions of copies of the red index oligo, the blue bead will have millions of copies of the blue index oligo, etc., across the thousands and thousands of beads that we have in our library. However, each bead also has what we call cell binding moieties on their outer shell that makes them physically sticky for the plasma membrane. So rather than needing to associate your cells with your indices by partitioning, you instead, just literally, to a given PCR tube, add however many thousands of cells you have in a particular sample, along with the corresponding number of these indexing beads. The cells and beads form one-to-one paired complexes driven by that cell binding moiety. And that allows us to get rid of the requirement for partitioning, but still allow for high fidelity one-to-one cell index association.
Why is it one-to-one? What prevents from?
It's a great question. There's a couple of things that go into having like one-to-one pairing rather than multivalency in either direction. One simple one is the beads are basically the same size as the cells. So the beads are about 15 microns in diameter, which is roughly in line with a typical mammalian cell that's used for single cell. And so keeping the size of the beads and the size of the cells in the same general regime biases heavily for like one-to-one pairing rather than multivalency in either direction, just due to steric effects. Like by contrast, you can imagine if your beads were like 1,000 times larger than a given cell, it'd be very easy for multiple cells to bind to a given bead and vice versa. So that's one thing, it's just the size. Another is more, we just empirically optimize the right ratio of cells to beads in a given size tube for high efficiency capture and for low doublets. Say for completeness, it's excess of beads over cells, because beads are basically free in our assay, whereas cells, you want to make sure you capture the cells effectively. But it's not like 100 times more beads than cells. It's within, generally speaking, the same order of magnitude. The last thing I'll say is we don't always, but we tend to do these cell bead pairing reactions in tubes that have a flat bottom. So like flat bottom PCR tubes or flat bottom plates. And literally what we do is we pipette the cells in, pipette the beads in, we quickly pipette mix up and down. The beads are magnetic, so they're heavy, so they drop very quickly into a monolayer on the bottom of the tube. The cells are a little lighter, so they follow like a minute later on top of the beads. As soon as a given cell like drops and falls on top of a bead and grabs hold of it, physically blocks any other cell from binding to that same bead due to the sort of monolayer on monolayer effect. So those three things stacked together is what gives us capture rates and doublet rates in line with partitioning technologies, even though it's just happening passively in a PCR tube.
Is the cell binding moiety specific to just mammalian cells, or what about insect cell cultures?
That's a great question. So we have two different chemistries for cell binding. Our universal chemistry is the one that comes sort of de novo with your kit, which has the generic affinity for plasma membrane. So it's not specific to any type of cell, nor to any species. If you'd be talking about things with like complex cell walls, it might be a little bit different, but generally speaking, for like higher eukaryotes, from everything that we and our customers have seen, it's a universal binding chemistry. There's also an alternative chemistry, which I have a teaser slide on, which is cell-specific, which instead of this universal membrane binding, instead has one or more antibodies that allows you to specifically target one or more cell types of interest without needing to do a separate FACS or MAX experiment. That's also something that is user customizable, so if there's interest in exploring that kind of selective cell binding, we'd be excited to talk. All right, so after that, you have however many thousands of cells in your tube, each of which has been paired to a different bead. You add a specially formulated indexing buffer and put the reaction on a PCR machine for lysis and then indexing. There's a couple of different things in this buffer, but the most important of which is a high viscosity compound, which slows the physical diffusion of nucleic acids in solution, which is what provides what we call kinetic confinement through the process of lysis and indexing. So to bring it all together, you add your cells and beads to a tube. They bind automatically, driven by the cell binding moiety. We add this viscous indexing buffer and put it on a PCR machine. There's a high temperature lysis step, which is fast and which lyses the cells, releases their nucleic acids, and also releases the indexing oligos from each bead. So where you used to have a single cell paired to a single bead, you then have a cloud of nucleic acids from that cell and from that bead. We then drop the temperature to an indexing temperature, which lets those indexing oligos hybridize to their cellular nucleic acids, which our first chemistry is just a 3' poly-A poly-T hybridization to the mRNA tail. Now, because this lysis and indexing takes place in this viscous buffer, providing kinetic confinement, each of these clouds containing the nucleic acids of a single cell bead pair is kept within a small, tight, confined volume, and those confined volumes are where the single cell indexing reactions take place. So at the end of this, you have thousands of cells, each of which has been indexed by a unique index oligo, but it's a reaction that's just done in normal PCR tubes, and it feels like setting up like an RT-PCR. Yeah.
Is there a thermostable enzyme then in this?
So subsequent to this is the library prep, like the RT-PCR. So yeah, so this is just the upfront indexing, and then it's a pretty traditional first strand, second strand PCR process.
You have the enzyme after this.
Exactly right, yeah. So the output of this step specifically is RNA from each cell with a unique index oligo hybridized to its tail that identifies basically the bead that it was paired with. And then you do a first strand cDNA extension from there. Okay, so this is the workflow. So it's a pretty fast workflow. It's just 8 hours to go from input cells to sample multiplexed and sequencer loadable libraries. There's a variety of pause points across the workflow. Two specific ones are highlighted here, though there's a number of other pause points. One pause point that I'll specifically call out is pretty early on. So after you pair your cells to the beads and then add them to that viscous buffer, but before you do the lysis or indexing or any of the subsequent like molecular biology, we've added a cryopreservative molecule to that viscous buffer, which lets you simply snap freeze your paired cells or put them in -80. And you can store them that way for basically however long you want, and you can do so without fixation, without any sort of covalent cross-linking. It's native cells, native nucleic acids, native proteins, and you can use that to basically separate your cell acquisition in the first hour and a half of the workflow from the library prep and the rest of the workflow. And you can take this out to several months and get basically no change in the single cell signal or biology. We have, I would say, pretty big majority of our customers taking advantage of this early fixation-free pause point. On one hand, just to you might get cells or samples like in the middle of the day or the afternoon or something like this, and you don't want to do a whole bunch of workflow like going into the evening hours. I would say close to 100% of our users use this early pause point just to like separate the specimen acquisition from the like whatever 6 hours of library prep thereafter. So on day one, when you're getting a particular set of samples, you'll do this bit, and then either the next day or whenever, you'll do the rest of the like 6 hour workflow. Some people will also use this to acquire like a relatively large number of samples over time, like say you're using patient samples or mouse samples or time point experiments. This allows you to acquire basically however many samples over time. Just freeze them as you get them, and then put them all together into a single batch to do the actual, single cell barcoding and the rest of the library prep. Oh, sorry, go for it. Yeah.
Yeah. So my question is, how tight is the binding of the beads to the cells, or if easily to be like it's separated? Or?
Oh, like if you freeze it, do you potentially lose the binding? It's very tight. It's a
It's by antibody antigen. If the cell is not intact,
so I'll say from what we have seen no material effect of the freezing process on the cell capture rate. For actually both our universal binding chemistry and our cell-specific binding chemistry, those are pretty strong cell bead binding events. And we don't see any change in cell binding when we freeze the cells versus leave them fresh. Reason for that is both of those binding chemistries involve a streptavidin biotin juncture as the key binding interface, and that's just very hard to reverse. Yeah.
Will you show data on how represented the transcriptome is being captured? Is it just the cytoplasmic mature RNA, or are you also capturing the nuclear RNA?
That's a great question. I don't know if in this deck I have specific like exonic intronic ratios. We would be able to follow up with you on that if useful. To a first approximation, we think it's a mix of basically just a mix of both. So one of the other things in that viscous buffer, other than the viscous compound, is a small molecule that at an elevated temperature, so at our lysis temperature, destabilize lipid membranes. And that'll be true regardless of whether it's the plasma membrane or the nuclear envelope. And so our expectation, and we can get you some data to sort of indirectly answer your questions there. Our expectation is that basically any lipid bilayer is more or less dissolved during that lysis step, including the nuclear envelope. But we can follow up with some of that. Great. So we launched our first kits commercially after a sort of extended early access with some of the most far-sighted genomics groups in the country. We launched our first kits commercially at ASHG in Denver in November. We have 8 and 16 sample kits on the market right now and 96 sample kits available for early access. I mean, this is all you need. It's basically this in a PCR machine and like ethanol. So it's truly a very accessible footprint, truly a very accessible workflow and kind of experiment to put together in the lab. Now to go through some data. So we and our customers have, in aggregate over the last year and a half, tried a pretty big spread of cell types, specimen types, species, things like that. Here's just a few from our own in-house efforts across PBMCs, dissociated spleens, and this one in the middle is actually primary neurons. So I don't know if there's many people that do CNS studies here, but typically speaking, if you're looking at central nervous system, you need to look at single nuclei rather than single neurons because it's hard to get dissociated neurons to work with other single cell technologies due to a variety of factors. The only real force that cells in our workflow experience is your pipette, basically. And so if you can pipette your cells without losing them or getting worried about aggregation or things like that, our technology is generally pretty agnostic as per the type of cell or specimen you're talking about. And it's a good example of one where most other single technologies may struggle on dissociated neurons, but our technology was able to handle this is just this might not be of huge interest to folks here, but some of our customers are like biotech or biopharma organizations that want to do relatively large-scale single-cell experiments. So this is something that we did in-house just to demonstrate the sample flexibility and scalability of our platform. So we had one of our R&D scientists do 4 96-well plates, so several hundred samples, of drug-treated PBMCs, and process them from cells to sequencer-ready libraries in a single day. Now this was a longer day. It was like a 10 or a 12-hour day compared with our normal 8-hour workflows. So you wouldn't necessarily want to do this every day. But realistically speaking, like once you've done the assay once or twice, a single user could realistically do like a full plate in a day. Or if you wanted to take advantage of that fixation-free pause point, you could spread an hour and a half on day 1 and then 6 hours on day 2, potentially up to 2 plates at a time. Anyways, both the simplicity and the price points of our assay, we think, make these kinds of studies generally approachable, even if, yeah.
And then what's some of the sparser boxes?
So those were the drugs killed the cells, and there were no cells. Yeah, so it's like this actually was like 5 or 10% of. This is just part of the samples here. This is the full several hundred samples. Yeah, so you can see. I mean, if you look through it, you can see some of the drugs will like ablate specific compartments. Others were basically just like cytotoxic. This is a pretty big and diverse set of 50 different drugs, and so some of them are just like cytotoxic, and they're incubated. I think here they're incubated overnight, maybe 24 hours, something like that. So it's long enough to have like a pretty material cellular response. As a general note, also for sounds like it's a little bit hand wavy, but we actually intended pretty genuinely as a company developing products. Like this is an accurate biology, right? This is actually what the biology is. Like some drugs are cytotoxic and they will kill the cells. And our hope is that with the right affordable, simple, flexible single cell technologies, you don't need to like obsessively pre-control everything up front. You should be okay to have single cell as like a sort of unbiased readout, and that includes readouts that maybe you don't go in knowing whether all of your samples or conditions are going to look the same. Great. So some data from customers. I'll say we used to have an R&D lab in Cambridge, England, and so some of our early customers were like neighborhood friends, basically, and so you'll see some of that in this slide in the next couple slides. So in the middle of the right here, just a couple of academic labs at Cambridge, one that's basically a hematopoiesis lab and one that studies neurodegenerative disorders. Actually, both of these labs for blood cell development and neurodegeneration respectively want to do what I call like maybe medium scale functional screening experiments. And so what they need is good in vitro models of their particular cell systems for blood malignancies and central nervous system cells respectively, but an ability to do hundreds of samples simultaneously with drug exposure or RNAi knockdown exposure, things like that. And both of these groups were really happy. And then this group here is a private life science company that wants to do very large scale immunodiscovery and immunoprofiling, but with like tens of thousands or hundreds of thousands of samples, so like considerably larger scales of samples than realistically approachable, we think, with other single cell technologies. And so these guys, because it's such a big project and set of work they want to do, they actually go real deep into our tech and its performance. And so this is part of their validation here. So on one hand, they looked at cell type clustering, making sure that in the right cell clusters, you see the right known transcriptional markers with there. They also cross-correlated our single-cell RNA-seq readout, which is traditional FACS and flow, to make sure on a per-sample basis you're getting a accurate representation of cell types across, and they find the correlation is quite tight. Never perfect, but it's quite tight. Did a variety of like robustness and reproducibility measurements as well. And they also compared our technology against a well-known droplet-based single-cell technology platform. It's worth noting that our technology, as I explained a few minutes ago, it works in a very different physical way compared with other single-cell technologies out there. But what you want to see is that it gives you basically the same biology. It gives you the same insights, it gives you the same figures that you can put into a paper or present at conferences or put into grant submissions. And this customer, like all of our customers, when they do a head-to-head, that's exactly what they find. So that's true whether you're looking at, for example, gross cell type or subtype discovery or the proportions that you find. It's true if you're looking at molecular measurements and looking, within or between different cell types or subtypes at differential expression or pathway enrichment or other things that may be of relevance just for actual biological insights. And so these guys were very happy to see this, and they ended up working with us for some very big projects that may be becoming public over time. Just the last bit of customer data. This is one of the other Cambridge labs. This is the one that does neurodegenerative diseases. And so basically what they wanted to do here is set up a system that would allow them to do large-scale functional screening in plates, but with a single-cell readout. This is something that they were basically planning on doing bulk RNA-seq until they found out about our platform and are now planning on doing this with single cell, because the price points are in the range where that's approachable for them, as is the workflow. So this is, I don't know if you guys know what pseudotime is. This is basically if you have a developmental trajectory and you look at different time points, this allows you informatically to sort of like approximate the trajectory of development, even if you're not looking at it in a specific functional basis. And you see from early time points to later time points, you basically see a set of discrete developmental stages here, 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 and here. And what they basically want to look at is this final developmental stage here. And even though in a given sample, like in a given tube, they don't have 100% purity, something that's nice doing analyses with single cell is you can specifically look at these cells. If you have a drug exposure or control or dose or whatever, you can specifically sub-select on a post hoc basis the cell types of interest and look for the activity or the pathway changes, etc., within those. And this allows you to do things where bulk analyses you would never be able to do. Last bit of data, so this is just some company internal data. As mentioned a couple minutes ago, we have a cell selective binding chemistry to complement our universal binding chemistry, which uses antibodies instead of the universal binding moieties. This is a very simple prototype experiment we did where we started out with a whole PBMC prep and then used beads with 3 different antibodies. Their pan-PBMC antibody or a T-cell or B-cell specific antibody respectively. And what you see here is when you do the binding with the pan-PBMC antibody, basically all of the main compartments of PBMCs light up roughly expected proportions, as you would hope. But for a T-cell specific antibody, you see a very strong, basically fold enrichment for T-cells, and conversely for the B-cell specific antibody, you see a very strong enrichment for B-cells. The data isn't as clean as you would get from like a good FACS or flow experiment. It's more similar to like a MAX or another bead purification kind of experiment. But given that this was also just like a one-off prototype experiment, we thought that this was a pretty exciting demonstration of the cell activity that can be programmed into this technology and done without actually adding a single step to the workflow. So this keeps within that same 8-hour cells to sequence or loadable library workflow, and in fact cuts down on the time just a little bit when you're using antibodies instead of a universal binding chemistry. Last quick couple points on this. We have a couple of people in our customer set that are interested in using this to, for example, do large numbers of samples, but they don't want to do a separate FACS or flow for each of those samples, so they're just planning on doing it in assay. We have other customers that are planning on using this kind of technology, not necessarily for that application, but where you have 1 or 2 cell types of interest that seem to be disappearing from your single cell data otherwise. Give a current specific example, microglia. Microglia is part of the central nervous system. They are a major therapeutic target for a lot of neurodegenerative diseases, but they are fickle cells. They can adhere to plasticware. They can lyse very easily if they're manhandled a little bit. And we have a fair number of people that are broadly across neuroscience that want to use our technology to specifically enrich for microglia, to do so without microfluidics, where that causes some challenges, to do so without using nuclei, because nuclei preps can also cause challenges. And this is basically a way of keeping your entire original crude sample intact and only binding the beads to the cells of interest. So it simplifies the pre-analytic sample processing and also allows you to zoom in on 1 or more cell types of interest.
You bring up a good point with the microglia. and neuronal samples, what if the cell dissociation process can't give you intact cytoplasmic membrane and all you get are nuclei?
Yep. So I should say we are coming out with a nuclei workflow in the next couple of months. So the same, as I say, our universal binding chemistry is basically a membrane binding chemistry. So we're putting some finishing touches on a validated nuclei workflow. We'll support nuclei for folks or applications where you want to do nuclei. So that's a great question. A second answer to that is, for most single cell technologies, you need to have pretty dissociated single cells, right? If you have a whole bunch of like clumps or debris or things like this, it doesn't just reduce the quality of your data, but often like you can clog the microfluidics with the droplet platforms. It's poorly dissociated specimens are generally incompatible with most other single cell technologies. Poorly dissociated specimens are not incompatible with our technology. There's nothing to clog. There's no particular liquid handling that would risk forming complex aggregates or things like that. And actually, for people that do difficult to disaggregate solid tissues, brain, CNS is a good example of that, be it primary tissues or be it like organoids. The organoids can be difficult to reliably and safely dissociate, as well as like myocardial tissues or other things like this. We're working with a couple of people on light or non-dissociated tissues as a modified version of our workflow. It'll subtly change the informatics that you want to do on the back end to make sure you're getting the exact right readouts that you want and it's the right insights that you're getting at the end of the day. But if there's an interest in either lightly dissociated specimens or non-dissociated specimens, we'd be very excited to talk because we think that's a big opportunity that our technology can address due to the fact that it's solution phase that other technologies realistically won't.
Is clumping behind why you get a few B cells in the T cell prep and a few T cells in the B cell?
It's hard to say. So we don't actually see much clumping in our tech. Basically, there's not that much liquid handling to go from the input cell specimen to the lysed and index cells. It happens just a couple of mimetic steps. Maybe we see a little bit of clumping, but generally speaking, it's a gentle liquid handling workflow between when you start and when you get the index nucleic acid. So there's not that much. I mean, truth be told, I forget exactly what these markers are, but it's not like there's no B cell markers on T cells and vice versa. And you can always get like a little bit of non-specific adhesion or stuff like that. Right. Yeah, Ryan leads our support organization.
So it can be, it's specific but not totally specific, and it's going to be based on the antibody that you use, which is going to be up to you. So you will get a little bit of carryover from other cell types in that case. If you have a really good antibody that's very specific to your cell type, it'll do a little better. But it's best to think of it more of as an enrichment, and it does a pretty good job, but it's not going to be 100%.
Okay.
All right, so this is just some quotes from a couple of our early customers. This group here is looking at doing quite large-scale organoid screens, including with automation, for example. So I'll say our technology is very automatable, almost regardless of the robotic platform or platforms that you may have. And some folks don't want to do quite large-scale experiments, or sort of organically thinking about automation just to make it reproducible and fast. Work with some of the folks at the Broad Institute on basically deploying this kind of technology at scale, be it in a core lab context or otherwise. We can't say much about this last one, but we were working with a corporate partner in the cell therapy space that, for a variety of reasons, wants to use single cell as a big chunk of their manufacturing pipeline. And we're looking at a variety of platforms, and for a variety of reasons, including the total, like fully baked cost, ended up going with us. So we were happy to see that. And we have a pretty big universe of customers across academics, translational core stuff, industry. Thank you. Okay. Time for questions. There's also food in the back. We could, I don't know if it's useful, we could quickly break for food and then Q&A, or however you guys want to do this.
I just was going to ask a quick question.
Sure.
So, cell viability is a big challenge in many single cell experiments. How is your technology forgiving for that? Like can't mRNA, all that.
For sure. I might toss this over to you, Brian.
Yeah. So we have procedures to clean up cells that may have low viability. So recommendations within the protocol. High viability is going to be a little bit of an issue because ambient RNA, you're going to have a lot of extra ambient RNA within the sample. So really it comes down to how well you are able to clean up those cells and get the debris out of there. Now, on top of that, you have low cell numbers. That's typically okay within our assay because it's easier to use those, to use samples that have low cell numbers and still have success within the assay. So, I mean, basically it's all in the prep of the cells, and then, and that's.
Do we have like a specific recommendation or requirement?
The recommendation is 85% or above, and it's not outrageous, but if you get a little bit lower than that, you can still get a little bit of data, but it may get a little bit noisier, and you're going to have dropouts in cells.
You said the barcoding is 3' end bias. Does that mean that it's a oligo DT based oligo? Is there one that's not oligo DT based that could maybe like capture viral transcripts that are not polyadenylated, or especially nuclear RNA is not going to have that poly A tail?
Yeah, so currently it's only polyadenylated transcripts that we detect. It's a poly-T oligomer that we use as the indexing oligo. So that's the only one that's available right now. And so for transcripts or other targets that aren't polyadenylated, will generally not be indexed or detected with our tech. It's not to say that we won't be coming out with products over time that are full transcriptome, but at present, no. One thing I'll say though, at least in terms of the polyadenylated transcriptome, is even though it's a 3' indexing event, we, due to the fact that all of our enzymatics is solution phase, we organically get pretty much full-length cDNA molecules. And then when we do the second strand cDNA synthesis, we down select those that are basically the right size as insert lengths put on a traditional short-read sequencer. So for folks that are interested in either 5' information or long-length or full-length transcripts for like isoforms or things like that, we're working on a couple of different projects with customers for those kinds of applications as well. It'll still be poly-A, poly-T focused though.
Yeah, but I'm interested in profiling like virus-infected cells, where some cells will be infected and many are not, and to be able to track which cells are infected and which are not would be really great.
Yeah, we've thought about that a little bit. One thing I would say is if there is any marker on the surface that could be a distinguishing hallmark of infected cells or uninfected cells, that using this sort of selective B chemistry would be one indirect way of getting at that, but it's not going to give you the intracellular hallmarks.
Yeah, that's what we want.
Yeah.
How about fixed cells? Fixed cells, or
right.
Yeah, so we're working on methanol fixation. But anything that's going to dry the cells out too much is going to be a bit of an issue. But we're looking for a partner if you want to do a methanol fixation study. We can run it for you guys.
So the main thing about fixation, we, as Brian says, like expect that some of the methanol or dehydration-based methods probably would be compatible, just because what you would want is to ensure that you haven't cross-linked the plasma membrane to such an extent that you can't permeabilize it during our lysis step. And for the methanol methods, which you can use for like fish protocols, for example, they're already basically permeabilized, so that sort of deals with that already. But some of the other methods where it would be tough to lyse the membrane subsequently for flow between, is probably harder. But yeah, we've had a couple customers ask about that, so if there'd be interest, I'd be excited to talk.
The tough part for us there is getting to the cells, so if you guys have some, you can go and chat.
Yeah. So another question is, so the B is going to be bind to the nucleus.
It's a great question.
If you cannot bind to nucleus, that means we have to use the fresh tissue or fresh cells.
It's a great question. We're coming out with a nuclei workflow. It'll be the same physical kits, but just a tweaked workflow for nuclei at some point, maybe two months from now, something like that, a couple months from
I'm
now.
hoping to go within that, yeah.
Yeah, within the next couple months. Yeah, and it'll be basically the same workflow, the same chemistry, but you'll have nuclei, a single nuclei suspension as the input.
But most of those transcripts and nuclei will not be polyadenylated. So
Yeah, so some of them are co-transcriptionally spliced and then polyadenylated, but also like the milieu of transcriptome in the nucleus is more complicated and a little bit different to the cytoplasmic transcriptome, generally speaking. So we would expect similar data to what you would get to any other 3' Jex chemistry when used with nuclei. Basically, is what we would expect. Which there's a non-trivial polyadenylated nuclear transcriptome. I mean, like superficially, like PBMCs, for example, just by volume are mostly nucleus or proportionally a lot of nucleus compared with the cytoplasm, and we get reasonable data from PBMCs. It's don't disagree.
all right.
Well, thank you guys. Appreciate it.
And we got some virus transcripts that well, we captured them in the clinic. We didn't expect to. So there might be some polyadenylation of the viruses because the literature says no. I'm going to stop this live stream.